Dear friends, good morning to all. Myself, Dr. G. Sivayalango, Assistant Professor, PG and Research Department of Biochemistry, Sacred College, Tirupattur. Now we can discuss about enzymology, enzyme kinetics, activation energy, enzyme catalysis, and factors affecting enzymatic reactions. First one, enzyme kinetics. Enzyme kinetics is the study of the chemical reactions that are catalyzed by enzymes. In enzyme kinetics, the reaction rate is measured and the effects of varying the conditions of reaction are investigated. Studying an enzyme's kinetics in this way can reveal the catalytic mechanisms of this enzyme, its role in metabolism, how its activity is controlled, and how a drug or a agonist might inhibit the enzyme. Next one, reaction rate. Reaction rate or rate of reaction is the speed at which reactants are converted to products. Next, move on to activation energy. The activation energy is the energy required to start a reaction. Enzymes are proteins that bind to a molecule or substrate to modify it and lower the energy required to make it react. And without enzymatic reaction means which needed activation energy low level. Without enzymatic reaction means which needed for activation energy more. Next one, enzyme catalysis. Enzyme catalysis is the catalysis of chemical reactions by specialized protein are known as enzymes. Catalysis of biochemical reactions in the cell is vital due to the very low reaction rate of uncatalyzed reactions. The mechanism of enzyme catalysis is similar in principle to other type of chemical reactions or chemical catalysis. The enzyme reduces the energy required to reach the highest energy are called as transition state of reaction. The reduction of activation energy, the activation energy indicated as EA, increases the amount of reactant molecules, the activation sufficient level of energy such that they reach the activation energy from the product. Next, move on to factors affecting enzymatic reaction or enzyme catalyzed reaction. The activity of enzymes is markedly affected by several factors. First one, effect of temperature. The rate of chemical reactions are increased as their temperature is raised. Rise in temperature which causes increase in reaction rate in enzymatic reactions. However, certain temperature commonly known as optimum temperature, usually in the range of 37 degrees Celsius to 45 degrees Celsius, above which enzymes being proteinous nature undergoes denaturation with losses the enzymatic activity. It's due to loss of secondary as well as tertiary structure of protein moiety. The graph shows the temperature, x-axis in temperature, y-axis in reaction velocity. X-axis different temperatures like 10, 20, 30, 40, 50, 60 as like when you are increasing the temperature, the reaction velocity increased certain temperature, after that it will be declined. As the enzyme is inactivated, the reaction which it catalyzes slow down ultimately stops. Low temperature generally do not destroy the enzyme. Its activity returns on heating to optimum temperature temperatures of 60 to 80 degrees Celsius permanently destroy the most of the enzymes. Most of the enzymes except fat hydrolysing enzymes are inactive at 0 degrees Celsius. Fat hydrolysing enzymes are found to be active at minus 50 degrees Celsius, and thus fat containing food may deteriorate when stored below 0 degrees Celsius. Thus, optimum temperature of enzyme may be defined as the optimum temperature at which its enzyme activity is maximum. The enzyme value of the optimum temperature depends upon the nature of the individual enzyme. The optimum temperature for most of the body enzymes under ordinary conditions is near the body temperature. Next one, effect of pH. The reaction velocity of an enzymatic reaction is very dependent upon the pH of the medium. For most of the enzymes, there is a range of pH, normally 4 to 9 values, outside which the enzymes will not function. Means most of the enzymes which are active at 4 to 9 pH. Optimum pH of an enzyme may be defined as the optimum pH at which enzyme has maximum efficiency or a maximum activity in the catalytic reaction. Some examples: optimum pH of pepsin 2, trypsin 8 to 9, urea is 6.8 to 7, salivary amylase 6.8 as a optimum pH. A typical relationship between reaction velocity and pH for a enzyme catalysis reaction is shown in this figure. X-axis different pHs, Y-axis for optimum activity of enzyme. The peak which indicates the optimum pH of enzyme. Third one, effect of concentration of enzymes. The velocity of an enzyme reaction is proportional to the concentration of enzyme. In case the enzyme concentration E is doubled, then twice active sites become available to combine with substrate provided and excess of substrate is present, and thus the maximum velocity is also doubled. Therefore, in general, V directly proportional to enzyme concentration. The graph also shows the x-axis for enzyme concentration, y-axis for enzyme velocity. Whenever we are increasing the enzyme concentration, the velocity also increased. Fourth one, effect of substrate concentration. With a fixed amount of enzyme, the reaction rate is proportional to the substrate concentration. But this is true up to certain concentration of substrate do not further increase the velocity of the reaction. The graph shows the x-axis for substrate concentration, y-axis for reaction velocity. Here we can consider 4 tubes, for example A, B, C, D. All the tubes having the 6 enzymes. 6 enzymes means 6 active sites only available. We are increasing the concentration of substrate. A only 2 substrates bounded. B 3 substrates only bounding. C and also D 6 substrates only bound here. Here we are increasing the substrate concentration. There is no active sites available, so that the saturation will be takes place. Next one, effect of concentration of product. Accumulation of product of the reaction causes a lowering of enzyme activity. This is prevented by removal of product from the site of formation. Example, absorption of the products of digestion is removed from the gastrointestinal tract into the bloodstream. Sixth one, effect of activators and also coenzymes. The activity of certain enzymes is greatly dependent on metal ion activator and also coenzymes, which induces the enzymatic activity. Seventh one, effect of modulators and inhibitors. When the active site is not available for binding of substrate, the enzyme activity may be reduced. The substances which stop or modify the enzymatic reaction are called as inhibitor or modulator. Presence of these substances in reaction medium can adversely affect the rate of enzymatic reaction. Last one, effect of time. The time required for completion of an enzymatic reaction increases with decreases in the temperature from its optimum. Whenever the optimum temperature, under the optimum or increased condition of temperature, which reduces the enzymatic activity. However, under the optimum condition of pH and the temperature, time required for enzymatic reaction is less. Means we are maintaining the optimum condition, means time requirement are very less. Thank you.